And hi, everybody. It's great to see everybody here. It's exciting today to have the chance to hear from Shani, who's, of course, now got her own lab at City University in New York, but she's been doing amazing science, for already so many years. She started out getting her PhD at Yale in biophysics and biochemistry, and then she did a postdoc with Cliff at Princeton, and then, of course, has set up her own lab. And it's of note that she's won a very prestigious career award already from the National Institutes of Neurological Disorders and Stroke to help her get the lab up and running. And she really is a rising star in this whole condensate field. Amazing accomplishments already. Publications on tau, alpha-synuclein, on the use of microfluidic methods to study condensates. She was the lead author on a very highly cited PNAS paper in 2015, which looked at LAF1 in P granules. She was the co-lead author on another 2015 paper in molecular cell, and Cliff was on that, and also Amy Gladfelder, that demonstrated the nature of these critical polyglutamine interactions with RNA, and also was important because it demonstrated that it was possible to fine-tune the material properties of condensates using RNA. And the last kitchen table talk speaker we had, Huaying, was also the other co-lead author on that paper. So obviously we all here think that's a great paper. She's also just been active in the field more generally. She's a co-author on a very important Nature Chemistry paper from 2017 that studied the role of IDRs in the function and behavior of that same protein LAF1. There's a really interesting trick in that paper, the ability to measure the mesh size of condensates and really getting at the whole length scale question of diffusion, very important problem. And now at SUNY, her lab, she's continuing, of course, to study phase separation, in particular with an emphasis on neurodegenerative disease and the aggregation of proteins in that space. And it's a wide variety of technologies that she uses: single molecule fluorescence, soft matter physics, genetics and C. elegans, obviously a strong emphasis on RNA granules. And she just continues to do great work and pump out really interesting results that all of us should really be paying very close attention to. So it's really an honor, Shani, to have you here today. And your talk is titled Emergent Material Properties of Biomolecular Condensates. So thanks for doing this. It's great to have you.
Thank you so much, Mark. That's really an incredible introduction. I don't even know how I can, move on from there. But thank you so much. It's really an honor to be invited and included in this really, really great series, this kitchen table talk series. And so today, I'm gonna hop around a little bit, but mostly speak about a recent paper where we use model condensates, but talk more broadly about our focus on emergent material properties and why we love them, why we care so much about them, and why we try desperately to measure them in many different ways. Oops, okay. So if there's ever a talk that does not need an introduction to condensates, it would probably be this one, hosted by condensates.com. And actually, I think it was a talk or two ago in the series that I believe it was Mark, and I hope I'm not misquoting you, who at some point jokingly said, well, all roads lead to condensates. And I think that's probably true, or at least seems true if you follow the literature. And so here, this is sort of now this famous picture, this pictorial summary of, which is already now probably rather outdated, but it was in this important review in 2017 that summarized in just, short, almost less than a decade, the variety of different granules, bodies, transit clusters of molecules that then was coined to be termed biomolecular condensates. And so I believe, this term condensates, right? So inherent to this is the understanding that it's really sort of an inclusive term, right? So sort of borrowed from the field of soft condensed matter physics. It's inclusive in the sense that some of these granules, at least initially, were shown to have liquid properties, such as the P granule in the seminal 2009 paper by Cliff and Tony Hyman, Cliff Brango and Tony Hyman. And then, of course, there's a variety of bodies now with a variety of different material properties. And so biomolecular condensates really is a nice inclusive term that takes into account this spectrum of properties. And so again, as the name implies, condensates are inherently defined by these properties, these emergent properties and emergent behaviors. So in the title, and I talk a lot about emergent behaviors, this is the word emergent, of course, right, denotes the fact that these features are novel. So the features that we see in these condensates are novel, and they emerge on the macro scale, on the scale of this new bulk network of interactions that occurs. And so, of course, a classic example from, again, the 2009 science paper would be the emergent property of wetting or flowing. And so here, these P granules that we see, originally they sit and they wet the surface of the nucleus. And upon shear stress, so this is in the adult C. elegans gonad, we can see these flowing and dripping and wetting. And this was one of the first indications that these P granules, since they have displayed this type of behavior, can be considered some sort of condensed state or a liquid-like state, as it was called at the time. Of course, in 2011, with the nucleolus, Cliff and Tony went on to show that that other properties, such as relaxation to spherical shapes, formation of liquid bridges, coalescence, again, sort of these emergent behavior were seen in the nucleolus. Emergent properties also give rise to novel local environments, right? So when you have phase separation that occurs, you now have a novel environment that is unique inside that condensate, which can only occur if there's a condensate, right? So this is an emergent property. And finally, multi-phase coexistence, first shown in the nucleolus in 2011, and now shown in several other systems in vivo, as well as in vitro for different proteins. This really also emerges from the unique properties such as viscosity and surface tension that give rise to this hierarchical organization of phases. So how phases that have distinct properties then or self-organize amongst each other in a system. Okay, so as I just mentioned, the term condensates or condensates, I guess, depending, I don't know if that's a 50-50 split in terms of pronunciation. I actually flip-flop between the two. I don't know why. So it's intentionally inclusive, I believe, and that denotes the fact that materials really exist, like many things, on a spectrum. And so on one end of the spectrum, you approach something that's sort of an ideal liquid or an ideal Newtonian fluid, which is defined always by this certain viscosity and a certain surface tension. The other side of the spectrum, you have sort of the idealized solid or Hookean solid, something that's defined by this elasticity, this constant elasticity. And then in the real world, you really have a spectrum of materials that fall somewhere in between, where unique blends of viscous behavior and elastic behavior can exist and coexist and or dominate at different length scales and under different conditions. So in my group, we're really obsessed with measuring and quantifying these properties. And we care about this for many reasons. Of course, for all the reasons I just described. So material properties are inherently, are inherent to the function and even just the definition of these things, these condensates. So not only in the way they wet or coalesce or fuse, but also in the way the internal molecules are organized, right? Material properties are also inherent in the discussion of pathology. So there's increasing evidence that's really sort of implying that changes in material properties. So the exact definitions of those properties, how something can go from something liquid-like to something solid-like, this process is being implicated in a variety of diseases, including neurodegeneration, which is one focus of the lab, which I'm actually not going to, unfortunately, really talk about today in this talk, but it's something that we care a lot about. And we also are really interested in understanding and quantifying properties from a design perspective. So we, understanding the fundamental principles that govern how molecular level interactions translate to certain material properties is incredibly useful in thinking of leveraging that information in the design or engineering of novel materials that are bio-inspired. And this is just a classic example of, the spider silk that has been just beautifully understood and engineered and even improved in bio-inspired models. And finally, perhaps most related to, the mission of Dewpoint Therapeutics is, we believe that quantifying emergent material properties is crucial for building therapeutic strategies to target condensates. And so, I really consider this to be, analogous to the way crystal structures have been invaluable in allowing for the development of targeted, drug therapies to understanding the three-dimensional structure, the three-dimensional map of a protein allows for efficient targeting. So too, understanding the three-dimensional network configuration of the condensate, I believe is going to be crucial for targeting condensates for future therapeutics. And so for these reasons, and because it's really cool and beautiful, which is implied, we study and measure material properties of condensates. And so one big thing that we ask, underlying all of these reasons is an approach or question we ask, which is, how do molecular level interactions give rise to these network properties that are novel that emerge from those interactions? And this relationship is actually not very intuitive or easy to predict. And so we're interested in really understanding that relationship. And so to do that, we do spend some time with reductionist approaches, both on the protein level, so maybe, a protein and nucleic acid, and then even more reductionist where we just use polymers to really try to go to the very fundamentals and say, what does one residue contribute to a condensate versus another? And so one of the methods that we use a lot that we really like is passive microreology. And so rheology, of course, is the study or science of material deformation and material response to force or stress. And in passive microreology, it's quite relatively simple. So in passive microreology, you don't apply any external stress or external force. You just leverage thermal fluctuations and you look at particle diffusion within a material. And so this doesn't really work for very, very, very solid materials, and it works for things that are closer to the viscous spectrum. And so here, these are just beads diffusing around in water. And so we understand the diffusion of beads, and if we track the particles and we plot the mean square displacement as a function of lag time, we expect this relationship. So the MSD will scale with the lag time with this exponent alpha, and alpha is equal to 1 if it's a pure viscous fluid. So if it's a pure viscous fluid in 2 dimensions, the mean square displacement is just going to equal 4dt, the diffusion coefficient. And in this case, we can actually extract the viscosity directly using Stokes-Einstein. Now it's interesting to note, so when you do have viscoelastic behavior, or in the extreme you have elastic behavior, you start to lose that linear time dependence. And so in the extreme elastic case, there's no time dependence to the bead motion over time. And so using this method, we can extract information about the viscoelasticity, and then we can explicitly define and quantify viscosities. And so just because, especially given the introduction and Hawaii's talk yesterday, I thought I would just do 2 little throwbacks from the early application of this method in condensates. And so, as Mark mentioned, I initially worked on developing this method and applying it to condensates back in 2015 as a postdoc with Cliff Brangwyn. And in this case, we were starting with just the P granule system, which at the time was just sort of liquid-like, looking down into the embryo, seeing it looked like a liquid. And I don't have time to talk about how fun 2015 or 2014, 2015 was in terms of trying to build those first in vitro reconstituted systems, right? Which proteins are involved, which ones actually form it. But long story short, we had identified the protein LAF1, which phase separates in vitro. It has an RGG domain, an arginine glycine rich domain, which was necessary and sufficient for phase separation. And we were able to quantify the viscosity of those condensates very reliably, and importantly show that that viscosity can be not only quantified, but then it can be tuned. And specifically, it can be tuned by addition of RNA binding partners. And in this case, just sort of short RNA polymers, in this case, make it more fluid and decrease the viscosity. Just after that, in that very same year, I had the real pleasure to work with Hawaiiing and collaborate with Amy Gladfelter and working on a different system, which is the We3 protein that we just heard about last month, I guess. And in this case, we show that not only can RNA tune the viscosity of a droplet, but actually the specificity of the RNA can specifically tune that viscosity, leading to condensates with unique material properties. And that's another really exciting area. And so LAF1, as I said, was driven by this arginine glycine rich domain. Long story short, it is now known that arginine glycine domains are really, really important. I couldn't list all the references here, nor all the proteins that have arginine glycine rich domains. This is just a few in a nice review from the Foreman K lab that talks about the arginine glycine rich domain. But a variety of P granule proteins, nucleolar proteins, stress granule proteins have an RGG domain, or sometimes called an RG rich domain, that is very important for phase separation and necessary for driving it. And of course, and now kitchen table talk, I don't know, a year ago or so from Rohit Bapu. If you don't know about the sticker and spacer model, this is a very important theme in the area of understanding the molecular grammar, right? So the grammar of understanding how sequences and how residues and their identity and their spacing are important for driving phase separation. And so arginine, of course, in this scenario is a sticker. So it engages not only in electrostatic interactions, but can engage in cation pi interactions and is showing up in a lot of phase separating systems. None the least of which are the ALS dipeptide arginine rich repeats, so these associated with the LS-C9 or dipeptide repeats that are arginine rich have been shown to alter both stress granule dynamics as well as nucleolar behavior and dynamics. So arginines are important in driving phase separation and are also implicated in maybe perturbing phase separation. So this is something that we became interested in, and specifically because arginines and lysines seem to show very different behavior. And so this is one example. There are several more now that show, so if for proteins that do rely on their arginine-rich domain to phase separate, arginine to lysine mutations are not conservative. So and this is an example from the Foreman K lab on FMRP, which is a neuronal granule protein. And when the arginines are mutated to lysines, you get differences in phase behavior. And recent work has also shown using FRAP and some coalescence that for dipeptide repeats, as well as short synthetic sequences, swapping arginine to lysine leads to apparent changes in material properties or emergent properties. So you get, better recovery from FRAP and faster coalescence for lysine than arginine. And so this is really fascinating, and I think there is a lot of literature even outside of phase separation that points to the unique differences between arginine and lysine, despite their conservation of charge. The distribution of charge in the guanidinium group, the arginine does give it some sense of some additional multivalency or additional versatility in interactions. And so we thought this was a great case to look at very specifically to understand and to quantify specifically the unique contributions of arginine and lysine, so we can understand again these principles from going from a molecule to a material, and how do these differences translate to these new emergent behaviors. And so in this case, we went really reductionist on this one. So we really just took homopolymers of polylysine and we took homopolymers of polyarginine. Of course, glycines are very, very important. And we hope to build up to that in understanding, but we wanted to look at this in just the purest way possible to compare these residues side by side. And so in this case, we have homopolymers of each, of increasing length, 10, 50, or 100, and an anionic binding partner, a poly-U RNA of either 10 or 50 length. And in some cases, UTP actually can also serve as an anionic partner, and in some cases, UDP. And so here's just an example of poly K100 with UTP. It forms these beautiful droplets. And everything, unless I state otherwise, at this point, since this is a complex co-acervate system, we just make everything charge match. So if we alter anything, everything is compared in a charge matched scenario. And so we apply our favorite technique. And so these are, I believe these are 500 nanometer beads. We actually use a few different sizes, from 100 or 200 to a micron or so. And we can measure the viscoelasticity. And so here, these are all of the beads that we track in this one very large droplet of polylysine. In this case, I believe this is poly K100 and UTP. And we see this slope of 1, so we can extract the viscosity. And actually, we can also plot the distribution of displacements for all the average over all of the beads over all of the in the system. And we see that these actually are Gaussian fits, which gives us another piece of information that this condensate is completely homogeneous. So every bead is in a similar environment. And this is consistent with idealized Brownian motion. Perhaps not surprisingly, when we increase the length of the polylysine polymer, so this is in the scenario of UTP, but we're just looking at polylysine 10, 50, and 100, we can increase the viscosity with length. And this is not surprising, but we were just playing with our system. And we can increase it both with polylysine length as well as increasing the anionic partner. And so we asked how this compares to polyarginine. And so before measuring the properties, we actually noted there were some important differences. There are differences in the phase propensity altogether. So in the case of UTP, where polylysine of each length formed very nice droplets, actually the polyarginine 100 formed aggregates, and that was the only case where we saw that, which was a difference. And interestingly, polylysine was not able to form condensates with UDP, but polyarginine was. Nice liquid droplets with UDP. And so this already suggests that the increased strength, interaction strength, and or an increase of valency of the polyarginine and the nucleotide interactions is giving rise to new behavior. So sort of, perhaps the polyR 100 and UTP, the interactions are so strong with the increased length, you actually get an aggregate and we don't see droplets at all. And UDP is not multivalent enough to form droplets with the lysine, but can form with the arginine. So perhaps the arginines contribute that valency. We don't have a model for this right now, but that's what we think. And then we can construct a phase diagram just to sort of, test what we would suspect, which is that the arginine droplets here in purple are much more stable in response to salt. So they can persist with higher salt. So the interactions are stronger and able to withstand higher salt than the lysine. Okay, so then we go to the rheology, which is the more exciting thing because we want to quantify things. We want to have metrics that we can compare directly. And so when we do this, it initially looks like the beads are not moving at all. The two movies are playing, but in the arginine case, we hardly see any bead motion. But you zoom in and you do see that they're moving, but just ever so slightly. And so what we do find, of course, we started this project expecting there to be a difference and hoping to quantify it. But what we were really surprised to see was how big that difference really, really was. And so the difference, this is all on a log scale. The difference that we see, so in the case of UTP between polylysine and polyarginine is several hundred, a factor of several hundred. And I think that anywhere between 100 and 300 higher viscosity for polyarginine than polylysine in situations where everything is otherwise perfectly analogous. And of course, this is a much bigger difference than just, increasing the length of the polymers. So this was, so something that was really surprising. And so, the polylysines, are maybe something on the order of a syrup, where it can flow, it's not too bad, but the arginines are really more like a paste or something very, very thick. And so at this point, this was actually meant to just be originally like a control of some sort, and we were going to move on to other things and build on this. But we were so interested in this big spectrum of viscosities, we thought this could be interesting from a design perspective, if to be able to span such a large range, could we mix or combine our lysine and arginine materials to form a more complete spectrum? So could we sort of occupy and make a sort of tune of viscosity range between these extremes, just using these few components? And the answer is no, we couldn't. Because whenever we did that, we either got exactly, no matter what combination we mixed them, we either got exactly the viscosity of the arginine itself, as if the lysine wasn't there, or the data looked terrible and we got nothing. And so one thing I should add, one nice thing about microbiology is that since we really only care about the particles and the particles are fluorescent, in these assays, we actually don't care. We don't need the protein to be labeled for these assays, or the droplet to be labeled, because we can see that with just DIC. And then we can see that the droplets are in there. And actually, that's really, really nice, because that's a good way to test for label artifacts. So we can do it with label and without. And that's just a plug. This is just a microbiology plug I'm making for some reason. But because of this, we thought, well, this is really strange. What's going on? And so we labeled our lysine and arginine. And I guess if you've already seen the social media plug for this event, you know that this happens. So this was really shocking, and this wasn't at all expected going into this project. The arginine and lysine, they do not mix. So the phases they're so different, and they're different enough, and they're unique enough that they actually form coexisting liquid phases, where here the arginine is labeled with 594, the polylysine is FTSE labeled, and the arginine is always nested within the lysine. And so initially, when we were trying to dial in all these different viscosities, either we were just looking at the arginine phase, which was always in the middle, and sometimes the lysine is just the shell, or we were looking at beads stuck at all these interfaces, and so the data didn't make any sense. And so this was beautiful, and I had this printed out on my desk for a really long time and just stared at it. I continue to stare at it. And again, sort of very surprising, and this sort of goes back to my intro statement, which is that these relationships are very hard to predict. So it's very hard to predict how you go from an particular amino acid to a viscosity. And it's even more so not necessarily known, certainly how and when you would get immiscible phases, considering these are both like charges, and they're both interacting with anions. All right, I made my point. This is exciting. Okay, so we know more information. So since the arginine is nested within the lysine, that already tells us something about the relative surface tensions. And so we wanted to test this by doing droplet coalescence experiments. And so here the arginine is not labeled, but the lysine is. And you can see that the arginine is fusing much more slowly than the lysine. And so this is quantified here. So polylysine fuses much more quickly than the polyarginine. And actually on the right here, the bottom right, we can actually compare the coalescence of the droplets when they're just alone or when they're in the mixed system. And that's symbolized by the circles and the crosses, and they actually align perfectly. And so, or somewhat perfectly. And so we believe that the properties in the separate phases are consistent with the coexisting phases. And this also suggests that the surface tension of the polyarginine is 10 times higher than the surface tension of the polylysine. And we would expect it to be in the nested within. Okay, so everything I've just showed you now is in this charge match situation, which I sort of predicated this part of the talk with. So there's enough anion to satisfy the amount of cation, even in the case where we add, the two polymers. And so we asked, well, what if we had different amounts, insufficient or excess? And so when we add excess, in this case is UTP. So when we add excess UTP, we continue to see the same thing. But interestingly, when we have insufficient UTP, so there's not enough UTP to satisfy the coacervation of both the arginine and lysine polymers, we actually only see the polyarginine droplets. And I guess, which is consistent with the fact that the arginine nucleotide interactions are much stronger than polylysine, and so it's really, out competing the polylysine. And then we thought, well, if they're really dominating the nucleotide interactions so strongly, what happens when we look at the system out of equilibrium? And so what we're doing here is we're making polylysine droplets with enough UTP to make nice, beautiful polylysine droplets. And then in this case, instead of mixing them all together initially and looking at them, we're adding polyarginine to the system and seeing what happens. And so, so this is essentially the limiting case once you add the arginine. So there's only enough UTP to satisfy, half of the total polymer. And so even though these polylysine droplets are already formed, once you add the polyarginine, you see them really just sort of going straight for the droplets, scavenging whatever UTP is there, and essentially releasing the lysine to the buffer and replacing the droplets. We call this droplet inversion. Very pretty. And so we can quantify this here. And so in the top, we can see that this, the slide coverage changes. So the lysine disappears and the arginine sort of takes over. And then in the bottom panels, if you just zoom in into one droplet, and this, it was apparent in the movie, we see that the arginine is actually nucleating within the lysine droplet. And actually on the right, what we can do is we can quantify the polylysine, the FTSE polylysine signal inside and outside of the droplet. And it's actually hard to see because, of course, once the lysine goes to the buffer, it's much more dilute. But if we quantify it, we, of course, see that we're getting this burst of polylysine out into the surrounding buffer as this process is happening and arginine is replacing it. So polyarginine is out competing the polylysine for the available UTP. And then we were curious in this case, so what about when the UTP is not limiting and even in excess in this case? So what do the non-equilibrium dynamics look like? And so here all these movies are playing at the same rate. And here we're repeating the experiment and we have either just enough or excess UTP. And so we know we end up at equilibrium with the same two-phase state. But the difference is that here, the kinetics of this inversion have changed. And so it gets slower and slower for 3 millimolar and 4 millimolar UTP. And then at 15 millimolar, there's so much excess UTP, we can't really capture it at this time scale. So we can actually quantify this kinetic. And so what we're essentially showing here is that increasing the stoichiometry of UTP is a way in this system to modulate the kinetics of this inversion, but also modulate the kinetics of the release of the polylysine into the buffer. So we like to think about the release aspect of it because we think from a design perspective, if this were built into a more complex system, could be a way to tune the release kinetics of some, say, compound or other molecule that associates with one phase versus another. And that's something that we're working towards developing or thinking about more. And so we also asked if we can tune this kinetic in other ways. And so because UTP concentration is a little extreme, what if we just change the length of what we were looking at? And so in this case, we're repeating the same experiment. Here, the only difference is that we're, instead of UTP being the anion, we're just changing to PU10 versus PU50. And so here, all of the concentrations are exactly the same, but just increasing the length of the anion in the system drastically alters these kinetics as well. And that is another interesting sort of viable knob by which to tune a system like this, which we thought was really fascinating. And that's just quantified here, is that we can play with these kinetics, and they can be increased or decreased. Okay, so I think timing. Okay, so I started by saying that these relationships are not easy to predict, and I think this story really highlights that, because we expected differences between arginine and lysine. We didn't necessarily expect them to be so different that they would be immiscible in most cases, and we found that we were able to actually control the dynamics of these coexisting multiple coexisting phases by playing with just out of the length or the concentration of these very minimal components. And so, this really lends insights into the unique roles of arginine versus lysine. And so let's go back to the situation where, arginine to lysine mutations alter phase separation. Well, you bet. I mean, based on this, you would really expect, based on the differences in phase behavior, that arginine and lysine would contribute to very different behavior. And we're not even considering post-translational modifications yet in this case. And that's clearly another way to build in complexity in this system. And additionally, we're thinking this can provide a framework for innovative use of arginine and lysines in designing tunable droplet systems that could be orthogonal. And so we're sort of interested in that as well from a design perspective. And so I'm just going to close with a summary again of sort of how we approach these problems in the lab. We measure these material properties and we try to understand these emergent behavior, both to understand function and dysfunction. I'm glad, okay, we didn't really have time to talk about pathology here, design, and then ultimately therapeutics down the line. And so this is the group. And so I just want to thank, so the bulk of this work was done by Rachel Fisher, a very talented postdoc in the lab. And her work was just featured. She won the first place in the Biophysical Society art contest. And this was using the same system, so compete or coexist, using the polylysine polyarginine system. And with that, I will take questions.
Great. Thank you, Shani.
Sure.
Wonderful lecture. So I think Jill wanted to solve this question once and for all. Is it condensate or condensate or condensate? So everyone has to vote. And we'll all vote while the questions are coming in. There's already a few.
Does it have to be constant?
Absolutely.
Okay.
It could be that, if we're talking about stress granules, it's condensate. And if we're talking about, P bodies, it's condensate or something. We can make it up exactly however we want to. So there's a couple of questions already that have come in. Maybe Jill, if you could unmute Karsten. Karsten has a really interesting question about the ability to reverse the behaviors you're seeing.
Yes, thank you. So really nice and clear talk. Thank you for that already. So I would have a question. So you said droplet inversion happens because the arginines basically have a higher interaction strength and compete basically for the polyanions. And I was wondering, could you reverse the process by adding excess of polylysine? Because typically coacervates are formed because salt or counterions are expelled, and you can reverse this process by adding excess of salt. So I was wondering, can you do the same with polylysine?
Yeah, so are you asking if we did the reverse? So if we add excess polylysine to polyarginine droplets?
Yes.
Yeah, right. Exactly. So yeah, we did that. I think it's in the supplementary. Nothing happens. So it doesn't work when you do it the other way. You eventually will get a two-phase system, but you don't get this. You don't get the inversion. So if you had preformed polyarginine droplets and then you add in polylysine, you don't see this.
Mm-hmm.
Very interesting. Yeah, thanks.
Good question though. Yeah.
That is interesting, and that leads to a really good follow-up that Charlotte had, and I had a similar question about spacers. So Charlotte, why don't you ask yours?
Yeah, thanks for a really great talk. I've enjoyed seeing all of these images on Twitter. And I was wondering if you had tried anything where you leave poly K as is and then introduce spacers into poly R so that you sort of dilute out the effect of the arginine, and if that could affect the extent of immiscibility.
Yeah, so no, that's a great question. So we've got a bunch of COVID stalled projects doing exactly that. So introducing spacers, but also oscillating between lysine and arginine in a single peptide to try to understand this and build the framework a little bit deeper into complexity. But that's TBD. But yeah, it's a great question. I mean, it's and this is sort of an extreme case where we have just the homopolymer, and certainly having the spacers in there. I mean, I guess I would speculate that having more spacers would also maybe allow for the two to be more miscible, right? So maybe the immiscibility wouldn't be as extreme, right? And you'd be able to have more flexibility, and maybe determining where that threshold is, and is something that we're interested in looking at.
Mm-hmm. That's great, and that that leads to, I think, another question that Anthony has about what you might expect to see in more complex situations. So Anthony, why don't you ask your question?
Right, so thank you for the great talk. I'm just curious, this was a really interesting study looking at well-defined condensates. But I was just curious how, I guess, the diffusion that you measure with your microreology measurements might change as you look at more complex condensates. Do you still get a single viscosity, or do you see heterogeneity within a condensate?
Yeah, it's a good question. So in our in vitro systems, we haven't gone more complex than a few components. And so far, in terms of looking at the homogeneity versus heterogeneity, like across the droplet, is that I think what you're asking? We don't really see heterogeneity unless there's maybe an aging scenario, and we see a progression towards a different state. And in some cases, there we do see some heterogeneity, and that's work that hopefully will be out soon-ish. So yeah, I mean, I think you definitely would get complexity as you increase components and as you increase, complexity of conditions.
Great, thanks.
Great.
Yeah.
So Deanna, you had a couple of interesting questions too.
Yeah, so I was wondering, that very beautiful talk, Shani. That was great. So I was wondering about the implications in signaling of your findings. So for instance, the nucleolus, it's highly enriched in arginine containing proteins. And then the nucleoplasm is highly enriched, or more enriched, in lysine containing proteins. And those are involved in transcription. So I'm just wondering just thinking of the nucleolus's role as a signaling hub, right? When under stress, it gets dissolved and a whole bunch of those arginine contaminants are released. What that effect might have on, dissolving the more labile transcriptional condensates or other.
Yeah, that's really interesting. So I don't even think I appreciated the fact that there are arginine rich and lysine rich things that are either separated in some situations or forced to possibly commingle in other situations. That is, yeah, I mean, I think this would have implications for that. It's hard to know exactly how without more details about the situation. But yeah, I mean, I think certainly, I mean, there's so many cases. I mean, even in the case of, histone tails, which are rich in arginines and lysines, even within similar settings. And so you can expect, yeah, I mean, I believe this would have implications for many of those systems. Yeah.
Thank you.
Great. So let's go to Jarell, because I think it's still interesting. All of us, I think, are still trying to wrap our heads around the idea that there's this immiscible, this property. I mean, yes, we all understand that arginine is aromatic and lysine is not. We know they're different, but still the fact that they're immiscible,
Right.
is just kind of weird.
Yeah.
And so Jarell has a really good follow-up question about that.
Yeah, thank you very much, Mark, and thank you, Shani, for a brilliant talk. I was wondering if you have a system that is, instead of made up of a homo polyaro poly K polymers, if you have a case where you have one polymer that is made up of 50-50 arginine, lysine, or like the first half is arginine and the second half is lysine, would the system be still immiscible, say, for example, for micelle-like architectures? Because I know that, well, I'm thinking that with this kind of scenarios, there would be a length dependence that may be factored into.
So that, I mean, that's literally what Rachel has been looking at now, is looking at sort of mixing these as maybe, in sort of a blocky, sort of co-block way, or interspersing the arginines and lysines. And so we don't really know yet, but we are doing some interesting things. And yes, since they are so different, well, we do suspect that there would be some really cool things that you can do depending on the the sort of separation or the clustering of the residues along a sequence. Yeah,
Thanks.
absolutely. Yeah, hopefully we'll see some. Soon.
Yeah, there's all these wonderful material science kinds of questions that you can ask, which may or may not have anything to do ultimately with condensates, but still, or condensates, but still, nonetheless, is just really interesting basic science. And so actually Patrick has, along the same lines, Patrick has a question about what happens if you start to introduce anions into the system. So Patrick, why don't you ask yours?
Yeah, Shana, lovely talk. Thanks so much.
Thanks.
My question is about adding additional polyanion species. So you've set up that if you have a single species of polyanion, that polyarginine can basically outcompete the polylysine for that. My question is, if you have two different species, so like, let's say poly U10 and poly U50, and then also the polylysine and the polyarginine, so 4 different polyanions, but everything charge matched. Mm-hmm. Do you expect that the polyanions would partition differently in the 2 phases? I'm assuming that there'll still be demixing, that you'll have 3-phase coexistence effectively. But my question is, can the arginine basically dictate which anion it gets?
Super interesting. So no, we haven't tried that specifically, but one thing related to that that we're interested in pursuing and hoping might be true is that, so, if we hope that certain molecules will preferentially partition in one phase versus the other, because then that would really open up really cool possibilities in terms of controlling, what is being stored, what is being released, and so if everything, assuming that there are differences between partitioning, then we could leverage that. But we haven't tried that specific experiment. So if there is enough for both, but maybe the interactions with the longer poly are strongest, and so those end up partitioning, would be like a possibility into the arginine phase, and then the shorter one would end up in the other phase. That's a really cool thing to try, actually.
Yeah, I think also somewhat relatedly, like still thinking about RNA, but just if you have the power to specify which polyanion you want, then going back towards a more biological system where you have structured RNAs, for instance, so more like the We3 poly and mRNA systems, then you could maybe start to get a sense of why you might partition one versus the other, depending on the charge content of the protein.
Yeah, and we follow Amy Gladfelter's work really closely in thinking about how RNA structure contributes to condensate properties, and we're really interested in that as well. And the other thing I'll just add, this is a sort of another ongoing project. So on the one hand, we can think of using the polymers to create different phases. We're also trying to see if we can use lysine and arginine as tags. So if we fuse them to other proteins, and then maybe that's where we get the specificity. So maybe we don't get the specificity from the polymer itself, but if the arginine and lysine can act as tags that create unique environments, but they're fused to something else that imparts the specificity, then that can also be very powerful and maybe more robust. And so that's something that we're looking at as well.
Sounds exciting. Thanks so much, Anna.
That's great. So Mike, you had an interesting question about the role of sterics.
Yeah, and I think I may have just found the answer to my question reading your paper really quickly, Shani. But no, I was just asking you in regards to the steric interactions of arginine, a bit more bulky than lysine. So it seemed that lysine would maybe pack a little bit more efficiently and therefore be maybe more viscous. Now, then maybe there's some role of like Laplace effects and Ostwald ripening and things moving around to make larger droplets with lysine versus arginine. But I think the answer to my question originally on the sterics, the kind of bulkier group of arginine, and tell me if I'm wrong or right, is that arginine has this kind of more delocalized structure and is able to undergo more pi-pi interactions and therefore creates a more viscous droplet. Is that correct?
Yeah, I mean, that's what we think. So we don't have any, modeling to really back this up. And if anyone wants to, look me up after this and talk about that, we can. I think that'd be really interesting to do. But we definitely think that's what's happening. And especially, the UTP, the UDP example, I think is also really telling, is that like UDP, we see this like all or nothing effect where like the polylysine just cannot, under the conditions we looked at, form anything with UDP, but the polyarginine can. And so, I mean, that would actually be really interesting to model more explicitly to try to understand that.
Very cool. Great talk.
Thank you.
Great. So let's go to Pradeep.
Oh, thanks for the great talk, Shani. I think you mentioned at some point that you were trying to tune the properties of tune the viscosity of the polycation and RNA system, and you sort of said you played around with like different combinations of arginine and lysine, and you found that the properties didn't vary much. Did I get that right?
So do you mean, so when I said that we tried to see if we mix them, could we get like intermediate viscosities? Is
right?
That,
yeah. So did you play around with the composition of a single polymer where you varied the lysine and arginine ratio, or did you just like mix poly R and poly?
That's what we're doing now. So yeah, so it was probably naive to think that we can just mix these and get out this intermediate like spectrum of viscosities. But perhaps by altering them just right within a single polymer, that we can achieve it that way. And so, right, I think, is that what you're getting at?
Yeah, I guess, and like maybe a follow-up to that idea, maybe potentially is that, so like most of your results show that like arginine seems to be interacting a bit more strongly with the RNA for whatever reason compared to lysine. And so if you have like
I mean, it's known in other like unrelated to phase separation or anything, it's known that arginine interacts more strongly with nucleic acids than lysine, and there's a host of other areas, including even just like antimicrobial peptides or things where like arginine can do things that lysine just can't, and for reasons that aren't well understood, other than that we know that there are essential differences, but in terms of there are a lot of functional differences that emerge from that, even outside of phase separation. Although if all roads lead to condensates, maybe all those processes also involve phase separation, but that's to be determined. All right, but back to your question.
Yeah, sorry. Yeah, I guess I was just just thinking out aloud about like if you have like a copolymer of like arginine and lysines, then you could sort of potentially play around with the relative amounts and like the patterning and such. And provided that if arginine's interactions are much stronger, then maybe you have to have like a much larger lysine to arginine ratio in your polymer before you start seeing an effect of the lysine RNA interactions itself as compared to the arginine RNA interactions. And
yeah, I think that's
so you could have some sort of like a steep decrease in the viscosity as you sort of like vary the arginine lysine ratio in your polymer. But yeah,
I think that's a sound hypothesis. I would agree with that. That it wouldn't sort of be linear in terms of ratio, and you would probably need more lysines to sort of break the arginine interaction effects. But yeah, but we'll see what happens.
All right. Thanks.
Great. So let's go back to Charlotte, who's continuing to think through very systematically this whole question of what we're learning about this, the balance here between what lysines do and what arginines do. Interesting question about aging. So
yeah.
Charlotte.
So I was sort of thinking the question that brought up the nucleolus, I was wondering if you had looked at aging, specifically if the components of the heteropolymers age differently than they would on their own. Like if the lysine component of the lysine arginine condensates, you know, sort of forms more of a gel faster than it would otherwise. And thinking sort of in a biological context, if this might be a way to explore how different enzymes might modulate interfacial regions to maintain each one's individual material components.
Yeah, that's such a great question. So one thing I'll say is that the polylysine droplets are just magical. So they just do not age, and they will just bounce off the surface, and then you can just resuspend them and they'll just form again. And it's unlike anything I've ever seen with the protein systems. And so yes, the polylysine are a beautiful example of like non-aging. I don't know if we've seen them age at all without possible evaporation or other issues happening in the sample. Whereas the arginine definitely does something. We haven't characterized really the aging of the arginine specifically, but you certainly can't do those same things with the arginine droplets. And so I think, yeah, I think you're definitely onto something. And that's a really interesting, an interesting thing to consider is not just the difference in material properties, but yeah, affecting the kinetics of potential aging. Yeah, but it's interesting. I mean, I don't know, and I'm just asking this question to you, to the audience. I mean, is it true? Like, so that's something to test. Like, so is it true that viscosity scales with the kinetics of aging? You know, I don't know if that's true, right? It's that simple, that things that are just more fluid inherently age more slowly than things that are viscous. And I think that, I don't know if that relationship is known to that extent, but it's interesting to think about.
Yeah, I bet it's a lot more subtle than that.
Yeah.
You know, and I bet it'll be so case specific. Yeah. Well, I think we've covered all the questions I see in the chat, and we're just about at the top of the hour anyway. So we'll have to continue some other time the debate over condensate versus condensate, although it appears that there is a bit of a preference for the first syllable being the stressed syllable, but a longer term debate, I think. So I would just like to, again, thank you, Shani. Wonderful talk, great questions, great participation from everybody. So thank you all very much. Wonderful to have your lecture.
Great, thank you.
All right. Thank you, Shani, and thanks everyone.
Thank you.
Okay.
Bye.
Bye-bye.
Bye.